Thank you for the opportunity to talk today. My name is Sarita Bennet. I'm an Associate Professor at Curtin University in Western Australia, and I'm a project leader on a project looking at the management of Sclerotinia in canola. So today I'm going to talk about increasing our understanding of the impact of management in canola to reduce the incidence of Sclerotinia stem rot. I'll start the talk by giving an background to canola production both in Australia and briefly as a comparison to South Africa. I will then introduce Sclerotinia stem rot and give a bit of a background about the life cycle of the disease, talk about its current management in Australia, and then we'll go on to talk about the work that we've been doing and along with our research trials, some of the results, finishing with some key messages and further research questions. Canola is the third largest broadacre crop grown in Australia after wheat and barley, with 1.9 to 2.7 million tonnes being grown each year. It's the major broadleaf rotation crop between the cereal crops, and we currently export over 2 million tonnes per year of our production, making Australia the second largest exporter in the world. You can see in the map below on the left-hand side that production is predominantly in southern across Australia, both in the west and in the east. This is our Mediterranean agricultural environments, making it very suitable for the production. In Western Australia in particular, in 2018-19 we grew an estimated 1.45 million tonnes, and which brings around $812 million per year to the state economy. So it's a very important component of the West Australian farming economy, and the vast majority of that is exported each year. Western Australia produces about 50% of the nation's production, looking at a 5-year average. Moving on to look at canola production between Australia and South Africa, you can see in the graph on the left-hand side that canola production in Australia started from very low levels in 2000, but has now increased to around 2 million tonnes per year in 2018. This is still significantly below production in barley and in wheat, but is still steadily increasing, although has plateaued over the last couple of years. The average yield in Australia is 1.5 tonnes per hectare, with some farmers reaching up to 2.2, 2.3 tonnes per hectare, but other farmers struggling to get over a tonne per hectare, depending on the conditions where they're growing their crop, particularly related to annual rainfall. In South Africa, production is on average around 100,000 tonnes per year, with the area sown in 2019 being 74,000 hectares. However, the average yield is not significantly different to that achieved in Australia at 1.3 tonnes per hectare. The major difference in South Africa is that you are currently importing about half of your canola oil that's required, and so there's significant opportunity to increase production. Now moving on to talk about the disease, Sclerotinia stem rot, or SSR, or Sclerotinia as it tends to be known. This is caused by the fungus Sclerotinia sclerotiorum, and it's distributed across all cropping regions around the world. It has over 400 hosts, which in broadacre cropping is predominantly the broadleaf crops, so canola, your pulses, lupins, soybeans, chickpeas, and also some of your other important crops such as sunflower in South Africa. It's also have significant damage in the horticultural industry, where crops such as lettuce can be reduced to a watery pulp. In Australia, it's estimated to cause up to $59 million per annum, but is highly variable as outbreaks are dependent on the environmental conditions. So in some years we'll have relatively little Sclerotinia infection, and other years you can have significant levels of infection. All of the current canola varieties are predominantly susceptible to SSR around the world, so there's a significant amount of work being done to develop varieties that show some resistance to Sclerotinia. The pictures on the right show you the disease cycle of Sclerotinia, as it's quite a complex disease. Starting with what looks like rat droppings in the picture in the bottom right. These are called sclerotia and are the melanised hard structures of the disease, which is where it over summers between the growing seasons. Then when the conditions are right during the winter growing season, they germinate these little mushrooms called apothecia, and it's the apothecia that release the ascospores that land on the petals of canola, and then as the petals drop, they tend to land in the leaf axis, which is nice and moist, and the ascospores, using the nutrients from the petals as they die, grow into the leaf axis and then into the stem. And looking at the picture in the top left-hand side, you can see the lesion of Sclerotinia just starting to grow. This then grows up and down the stem, and in severe cases of Sclerotinia causes complete destruction of the plant because it's stopping both moisture and nutrients moving up and down the plant. As the plant dies at the end of the season, the Sclerotinia forms the hard melanised structures again within the stem, and as the plant is harvested, the stem is broken and the sclerotia fall down onto the soil surface, starting the cycle again. So moving on to management in Australia, the main management methods being used are either canola breaks, where you have alternative plantings between years of canola, burning the stubble, as you can see in the graph picture in the right hand side, moldboard ploughing, where the sclerotia on the soil surface are buried, or we've got the option of chemical foliar fungicides. All of these have mixed success, and with increased canola plantings and long-term sclerotia inoculum build-up in the soil, this is putting pressure on traditional control techniques. So the sclerotia can last up to 7 years on the soil surface, and if canola rotations are more frequent than this, which they frequently are in Australia, moving now often to a one-on-one rotation, then there's significant opportunity for the sclerotia to build up. Burning the stubble works very well when it's windrowed in the windrows, but that still leaves numbers of sclerotia outside of those windrows which are not controlled by the burning because the temperatures aren't high enough. Moldboard ploughing may bury the sclerotia, but depending how frequently that moldboard ploughing occurs, you can bring them back up to the surface again, and there's been very little research looking at how long the sclerotia lasts when they're buried, and therefore whether they do degrade over time. Foliar fungicides are highly dependent on one, accurate prediction of ascospore release and the presence of the ascospores on the floral tissues, but also on the specific environmental conditions that are vital for disease development. So you're not only predicting when the spores are present on the floral tissues to control them with the fungicides, but if the environmental conditions are not conducive to the disease, then the money spent on applying the fungicide may be not economic. And therefore it's essential to understand the within-season risks in the canola Australian growing season to determine the efficacy and also the economy of foliar fungicides, and whether there's other management options that can be used that will help us to control Sclerotinia. So I'll now go on and talk about some of the work that we've been doing in Western Australia, and I'm going to concentrate on some trials that we did in 2018. This is because the trials in 2017 we only had a couple of varieties, and in 2019, although we had more varieties and we had them spread across Western Australia, we actually had no disease, so the conditions were not conducive. So 2018 we had a mixture of both, so I'm going to concentrate on those. So in 2018 we had 10 field trials from Geraldton in the north down to Cranbrook, which is north of Albany in the south. Each field trial had 2 open pollinated varieties and 2 hybrid varieties, with the varieties chosen being those that are the dominant varieties grown in the state. and each of the trials had a plus or minus fungicide which was applied at 30% flowering, and 30% flowering is used because this is the time when Sclerotinia is most at risk of occurring in the crop and is the most likely time when you've got maximum spore release of the ascospores. At each site we also recorded temperature, rainfall and humidity, and they were recorded right through the growing season. And then at each site through the growing season we've completed a detailed assessment risk for Sclerotinia. Now, the detailed assessment risk was done to determine the impact of previous Sclerotinia infection on in-season SSR. So the sites were soil sampled prior to seeding, where we sampled soil at each different plot so that we were able to see whether there was an increased risk in some plots of higher infection because of sclerotia that were on the soil surface. Petals were also collected at 30% flowering from all of the non-sprayed plots to determine whether the infection risk was coming from ascospores on the petals and therefore not from the site but could have blown in from other sites as well. We also collected disease incidence and severity within each plot, and then at the end of the season prior to harvest we collected 10 infected stems from each plot, and these was recorded for a lesion length, sclerotia number within each lesion and sclerotia size. The reason that we collected the soil survey results was based on research that we conducted in 2017, which followed a severe infection in 2016. In 2017, we soil sampled 6 different sites across the wheat belt, picking a mix of sites where we knew there was bad infection, so sites 2, 3 and 6 in 2016, and comparing those to sites where there was much lower levels of infection. And as you can see from the results in the graph on in the left-hand side, there were significant numbers of sclerotia left in the soil in the sites where there was Sclerotinia infection. So it does severely increase the risk of getting Sclerotinia in subsequent years, but it was highly variable depending where the Sclerotinia had occurred the previous year. And of course the spores that have germinated from the apothecia then have the potential also to blow into adjacent fields. So when there was high infection in the paddock 3, for example, paddock 4 would also be at high risk because of being so close. So providing a break crop within a paddock between canola isn't necessarily the only management option that's required, because paddocks that are adjacent to those that have had bad infection are also at risk in future years, particularly if they're in the direction of the prevailing wind. In 2018, I've just presented here the results from 2 sites because they were the only 2 sites that had significant numbers of sclerotia. The site at Greenough at the bottom shows that there was quite significant numbers per metre squared, with up to 16 in one plot, but it was also highly variable, with some plots having no infection or no sclerotia left on the soil surface. Bulgut was a much lower level of sclerotia, varying from 10 down to 0 again. So it's highly variable. Petal testing, as I've said previously, was done at 30% flowering because this is the time that spraying is recommended, but also this is when maximum ascospores are thought to be in the air and landing on the petals, which are also starting to drop at 30% flowering. And the way we did this was to collect 12 petals randomly throughout each plot, selecting only from the unsprayed plots, and these were then grown on agar plates so that we could record them for mycelial growth. And you can see from the graph on the right hand side that infection levels across all of the sites in 2018 varied between 60% and 100%. So we had significant numbers of ascospores present on the petals, and therefore the potential risk of infection of Sclerotinia in 2018 was quite high. However, the disease incidence at the end of the season was actually much lower. So The graph on the left-hand side is showing you the results between sites, where you can see the difference between the sprayed plots and the unsprayed plots across all of the different varieties. Bollgard was the site that had the highest disease infection, with up to 20% infection in some plots, and was significantly different from the unsprayed plots. Other sites had much less infection levels, so the conditions either weren't conducive or and therefore didn't lead to infection. As we saw from the previous graph, the potential for infection was there because of all those ascospores on the petals, but it hasn't actually led to infection at the end of the season. If we look at the graph on the right-hand side, this is actually done looking at the different varieties, and you can see that there does appear to be some difference between varieties, with Mako being more susceptible than others. Mako is an open pollinated variety and has very thin stems, but there is some difference between the sprayed and the unsprayed plots in the amount of Sclerotinia infection recorded in each plot. However, how much disease is actually required to compromise final yield, and which then leads to that question of when is it economically beneficial to spray? And that's the hard thing to know, because you have to spray before you see the infection. You've got to make that decision of whether you're likely to get infection, whether the weather conditions are right, and whether you're going to get a yield penalty if you don't spray. But also the more important question is how much sclerotia are you also going to contribute to following years? It's not just thinking about canola as an annual crop and managing that crop within that 1 year. It's also thinking about potential infection in future years as well. And as you can see from the results here, that despite having up to 20% infection in Bollgard in particular, there is no significant difference at all sites between the sprayed and the unsprayed plots. And also when you look at the different varieties, you can see again that there's no significant difference between the sprayed and the unsprayed plots within each variety. So although Mako appeared to have much higher levels of Sclerotinia in the unsprayed plots, it hasn't actually resulted in a significant yield difference. And the yield differences that we can see here are indicative of the yield differences that you would see between OP crops and hybrid crops, with the yield being higher in the hybrid crops. So the key messages from 2018 are that the greatest risk of Sclerotinia infection in canola field trials is from the airborne spores on the petals, that the sclerotia on the soil surface we saw in the majority of the trials was very low, and that we didn't have much infection that was a risk coming from those sites. Hybrid varieties are not necessarily more susceptible than the open pollinated varieties, and in fact Mako appeared to be the variety that had the most infection in 2018. This hasn't actually carried through into other years, so in other years we've had other varieties appear to be more susceptible, which is why we're now going on to do some more detailed work in the glasshouse looking at infection potential in varieties where we're actually controlling the conditions, so we can actually have a definite comparison between varieties. And that infection is highly related to the environmental conditions. So if you don't get the right environmental conditions, then it doesn't lead to infection, even though that risk of infection is very high from the ascospores on the petals.
So to finish, if we can increase yield by even 1% through a better understanding of the factors affecting Sclerotinia infection of canola, which will lead to improved management of the disease, this has the potential to lead to benefits for WA growers that will be close to 1.5 million per annum, so a significant saving. In order to achieve this, there's a number of key questions that we need to be investigating further. What level of disease is required to lead to a yield reduction in the crop? How do we know when it will be a bad infection year so that when it will be economic to spray? And is there really any difference in the resistance between varieties? My research group is currently investigating each of these questions in more detail, and we hope to provide answers to these questions in coming years. Thank you very much for the opportunity to talk to you today. And I understand that if you have any questions following the talk, that you can email those through, and then I will have the opportunity to answer those questions. Thank you.